Hello students, I am Dr. Sonal Gupta. I am working as an assistant professor at K.L. Mehta Dayanand College that is affiliated to MD University Rohtak. So here we will start with the module that is the development of different biopharmaceuticals using cell culture techniques, and the learning objectives of this module are what are biopharmaceuticals, their different types, their market status, and lastly the different techniques which are used for the development of biopharmaceutical products. That would include microbial cell systems, mammalian cell cultures, plant cell cultures, animal transgenics and plant transgenics. Let's start with the topic development of biopharmaceuticals using cell culture technology. First of all, we need to understand what are biopharmaceuticals. As a definition, biopharmaceuticals are the therapeutic agents of biological origin where recombinant DNA or hybridoma technology has been applied. Biopharmaceuticals are the recombinant therapeutic proteins derived from biological sources such as tissues and organs, microorganisms, animal fluids, genetically modified cells or organisms. Next, we will cover the cell culture techniques. With the advent of recombinant technology, the research and development of many biopharmaceuticals has enhanced, and we see many biopharmaceutical approvals as therapeutic products in the market. Biopharmaceutical products are defined as recombinant proteins having therapeutic effect or monoclonal antibodies which are produced via culture of recombinant mammalian cell lines at large scale, hybridoma technology and phage display. The different categories of biological products consist of hormones, enzymes, synthetic peptides, oligonucleotides, vaccines, gene and cell therapy products, blood and blood derivatives and polyclonal antibodies. The first biopharmaceutical in this category was human insulin that was first developed by Genentech and marketed with the name of Humulin by the company Eli Lilly. Since 1986, the human genetic coding for proinsulin is inserted into E. coli cells, which are then grown by fermentation to produce proinsulin. The connecting peptide is cleaved enzymatically from proinsulin to produce human insulin. Therefore, the production of human insulin through recombinant DNA technology represents an important advance in the treatment of patients with diabetes. And the same has been explained here. In this diagram, we can see how human insulin was produced first from E. coli cells for large-scale production of this hormone. Now, we will explain something about the market estimate of these biopharmaceutical products. Around 100 recombinant technology-based proteins have been approved by regulatory agencies worldwide, that is like by US FDA and EMEA, and these numbers are on rise. With number of these innovative products going off patent, there is a push towards approval of biosimilars of these products. As a consequence of this growing demand, the expression systems, mostly the cell lines from where these proteins are expressed, need to be produced at higher and faster rates. Now, we will cover the next learning objective of this module, that is how to prepare the different types of biopharmaceutical products using microbial fermentation system, mammalian cell cultures, plant cell cultures, animal transgenics and plant transgenics. A recent study on analyzing the supply and demand for mammalian cell culture capacity for biopharmaceutical manufacturing showed that technological advances in manufacturing process have improved the production levels of these products. This continues to result in significant changes in the supply and demand for mammalian cell culture capacity. New trends would be dispensed in the coming years which will enhance the growth, availability and utilization of biopharmaceutical manufacturing. Some of the factors which have led to improvement in production levels of these products are improvements in expression levels, widespread acceptance of single-use bioreactors, emergence of biosimilar development and consolidation in the biopharmaceutical industry. To achieve the objective, one needs to Number 1. Select an expression system or cell line which expresses the desired molecule at high rates with possibly low operational cost. Number 2. Culture media selection and optimization of culture conditions for higher yields. Number 3. In-process quality control for assessing compliance during manufacturing. And lastly, the overall quality control to meet the specification criteria. Now, we will discuss the first system, that is the microbial system. Microbial systems are an attractive source for production of certain biopharmaceutical proteins as they provide certain advantages like high productivity at low operational costs and rapid execution. Also, for the concern of regulatory authorities, there is no adventitious virus present. Some of the simple recombinant proteins like insulin has been successfully produced in bacteria E. coli. For the production of antibody fragments, E. coli was used, which is then pegylated so as to decrease its in vivo clearance rate. Also, the whole antibody, which is non-glycosylated, can be produced in the E. coli system and can be secreted extracellularly. Microbial systems have certain advantages over animal cell lines in terms of cheaper maintenance, easy handling, shorter production cycles, and much higher productivity. The major drawbacks of using microbial systems is limited expression of proteins as they lack post-translational modifications, assembling of multiple protein subunits, presence of various cofactors, and many more. Certain recombinant proteins such as tissue plasminogen activator and erythropoietin and certain monoclonal antibodies need glycosylation to retain their biological activity. However, these challenges can be easily overcome using animal cell lines for the production of biopharmaceuticals as they have well-defined machinery available for such modifications and assembly. The next system is the mammalian expression system or the cell lines. For the clinical applications, mammalian cells have become the dominant system for the production of recombinant proteins because of their capability to properly fold the proteins and add human-like post-translational modifications. The cell lines that are being used for biopharmaceutical protein production so far have been originated from mammals. Most commonly used cell lines for the production of recombinant proteins and monoclonal antibodies have been the following animal cell lines. Their capability of high productivities, optimized protocols, and ease of handling has led to their widespread acceptance and use by biopharmaceutical industry. Some of the cell lines used are Chinese hamster ovary cell lines, murine myeloma cell lines, baby hamster kidney cells. In case of non-human cell lines, there are chances of some post-translational modifications which are non-similar to humans, as a result of which immunogenic reactions can be triggered. And this is the main reason for choosing human cell lines. Therefore, the protein produced generally results in post-translational modifications similar to humans. And the same can be explained in this diagram where the production of tissue plasminogen activator is shown by using mammalian cell lines. The advantage of using human cell lines for biopharmaceutical production is their ability to produce similar type of proteins which are chemically similar to the one produced by human organelles. The structure, position and number of post-translational glycans is similar to that of proteins produced by humans, and thus their activity, immune response, body clearance and most importantly among them, their stability is maintained using human cell lines for therapeutically important protein production. The first important breakthrough in establishing human cell line was made in early 1950s by establishing HeLa or human cervical cancer cell lines. Within a decade, successful vaccine production was achieved using human-derived diploid cells. With early objections and concerns, currently these cells are successfully used for production of various viral vaccines. To achieve high yields for production of therapeutically important recombinant proteins and various monoclonal antibodies, human or mammalian cell lines are first choice for low investment, large scale production optimization. With advancement in the field of technology, various human or mammalian cell lines are developed for high production of therapeutically important biological derived recombinant proteins, vaccines, etc. Some newly established cell lines include fibrosarcoma HT1080 cell line, human embryonic kidney 293 cell line, while certain cell lines under awaited approval which have shown promising results include kidney B cell hybrid, hepatocarcinoma cell line, retinal cell line from 18-week fetus and human amniocytes derived cell lines. Major challenges in using mammalian cells are the cells are fragile because of their large size than microbial cells, which is approximately 10 to 50 folds higher. Mammalian cells lack the tough cell walls like microbes and are also sensitive to minute impurities. Therefore, it is a challenge to cultivate these cells in a culture for large-scale production of proteins. Moreover, development of mammalian cell lines is also very time-consuming, and to develop commercially viable processes, stability of protein expression is very important. So, it is critical to develop the cell lines that can secrete the protein of interest with high specific productivity and at high levels with unlimited number of cell generations. This will allow for a scale-up and cost-efficient manufacturing. To achieve this goal, using expression vector systems and cell line engineering could be used. The very common techniques used for biopharmaceutical production includes recombinant DNA technology and hybridoma technology, where choice of recombinant proteins including various hormones and other proteins are produced along with various other byproducts or derivatives of existing natural proteins such as immunotoxins, fusion proteins and vaccines. The applications are not restricted till protein and vaccine production, rather various vector systems like viruses and plasmids, small interfering RNA mediated gene therapy and various monoclonal antibody productions can also be achieved. Critical factors involved in this process include cell line transfection and selection. To develop a cell culture process, it is important to select the cells having the ability to produce and select for a high producing animal cell line. Some cell lines like CHO cells that have become the standard in the production of recombinant proteins. Some of the alternatives present are the mice myeloma cells, baby hamster kidney cells, human embryonic kidney or human retina derived cells. These cell lines are listed above and are adapted to grow as suspension cultures, therefore are suitable to be scaled up in the stirred tank bioreactors. These cells also bring an advantage to the industry because of the presence of well-characterized technologies that allow for transfection, amplification and selection of high producer clones of these cells. These cells can easily be transfected with the desired gene along with an amplifiable gene like dihydrofolate reductase or glutamine synthetase that offers an effective platform to express the required proteins. To select the positive clones using these systems, cell cultures are applied with a selective pressure using an inhibitor of DHFR or GS enzymes, which leads to increase in the copy number of transfected genes including the desired or recombinant gene. The first system is DHFR system. To use the DHFR system routinely with CHO cells, the cells deficient in the DHFR activity are used. Plasmid expression vectors are used to deliver the desired gene in the cells along with the DHFR marker gene. The expression vector containing the strong viral promoter is used to drive transcription of the recombinant gene. The vector is delivered inside the cells using non-viral transfer techniques. The techniques generally used for transfection are calcium phosphate, electroporation, lipofication, or polymer-mediated gene transfer. To select the transfected cells, a medium that requires the activity of DHFR for nucleotide synthesis and cell growth is used. To promote the amplification of the desired gene, the cells are exposed to a number of rounds of progressively increased concentrations of DHFR enzyme inhibitor, methotrexate. Methotrexate treatment enhances the production of a specific protein following an increase in gene copy number to several hundred in selected cells. The next system is glutamine synthetase expression system. The glutamine synthetase expression system is an alternative of DHFR system. GS system works as a dominant selectable marker. This is advantageous because there is no requirement of using any specific mutant cells. NS0 cells are preferred over CHO cells for this system because of the absence of glutamine synthetase. For the purpose, lower activity of an enzyme inhibitor can be used for selection and amplification in NS0 cell lines. Now we will study about the culture modes. It is a critical parameter to ensure the stability of selected clones for long-term culture, and for the purpose of commercial production in case of DHFR system, methotrexate is used to apply the selective pressure to select the positive clones and causes gene amplification. But a percentage of these genes are unstable, and by removing the selective agent which is necessary in production cultures results in a steady loss of the gene copy number. Reduction in gene copy number will lead to loss of production. There happens to be a loss of messenger RNA for the recombinant protein over long-term culture. Therefore, selecting the clones for high levels of recombinant messenger RNA may be useful as a predictor of a stable protein production. To get the higher producer clone, integration of the expression vector in the host cell genome is required. The site where the gene gets integrated in the genome of cell has the major effect on the rate of transcription of the recombinant gene. Also, progressive gene silencing can occur over successive culture passages. This is associated with the spread of heterochromatin, which is condensed and transcriptionally silent. Using expression systems, it causes random integration of transgene into the host cell, which leads to variability in its expression and stability. To prevent gene silencing, ubiquitous chromatin opening elements have been added into the vectors to prevent gene silencing and give stable, consistent and high-level gene expression irrespective of the chromosomal integration site. The next point is the stability of gene expression system. The cells can be grown in batch cultures to above 10⁶ cells per ml over 3 to 4 days to allow synthesis and product secretion. But the growth and production are limited because of the accumulation of metabolic byproducts such as ammonia. There may be depletion of nutrients such as glucose or glutamine, which can also lead to growth reduction. To address these limitations, the cells can be grown in perfusion cultures where constant supply of nutrients is there and the removal of media leads to cell densities of at least 10⁻10 cells per ml. The strategies that are proven to be most successful involve preparing feeding concentration of nutrients based upon the predicted requirement of the cells for growth and production. This involves slow feeding of the key nutrients at low concentration. The maintenance of low concentration of carbon substrates enables efficient primary metabolism, which leads to lower rates by product production. This results in longer productive state of cells over extended time periods. Fed batch cultures are mainly used to get higher yields using this strategy. To grow animal cell cultures, normally stainless steel stirred tank bioreactors are used that are designed with impellers to minimize shear forces. Producer cells can be grown in this environment by providing with suitable growth media and controlling gas sparging. The capacity of commercial bioreactors for animal cells is up to 20,000 liters from some of the larger biopharmaceutical companies. For large-scale animal cell culturing, airlift bioreactors have also been employed and have been shown efficient for protein production. Perfusion cultures are more demanding for large-scale cultures because of the potential advantage of allowing a continuous stream of product over several weeks or months. It is also advantageous because of the rapid removal of labile products from the culture. An effective cell separator directs the protein-containing media into a chromatography column, which is suitable for extraction and downstream processing. Another advantage of this mode of culture is that a 10 times smaller bioreactor can be used for the production of the same quantity of product. Bioprocess design became of greater importance when some of the first generation blockbuster drugs, example erythropoietin, human growth hormone and alpha interferon, start being produced as generics. To ensure the success of animal cell culture processes, several challenging areas of bioprocess development are required to be addressed, which mainly include serum-free media, apoptosis, glycosylation. So, here we will discuss about the serum-free media. From several decades, bovine serum albumin was used as additional supplement in the cell culture medias as a swift promoter of growth because it is rich in hormones, growth factors and trace elements. The high albumin content in BSA protects the cells from adverse conditions like fluctuations in pH or shear stresses. Although BSA has all these benefits, but the major drawback is that the composition serum is always variable and indeterminate, leading to unpredictable and inconsistent growth and productivity. This is a great challenge to identify effectual substitutes for all the growth promoting factors that are there in the serum. The requirement for serum varies for different cell lines due to difference in their growth patterns. This is why it is impossible to design a particular serum-free formulation which can act as a substitute for serum and is suitable for the growth of a cell line. Even different clones of same cell line may require different formulations for their optimal growth. Various strategies can be used to devise serum and animal component-free formulations. Like, in some cases, metabolic analysis of cells may help in media design. The third main factor is apoptosis. The main factor leading to the termination of cell cultures is apoptosis or cell death. The cell death is usually caused by factors like nutrient depletion in cell culture medias, metabolite excretion, too much shear forces. Necrosis cell death can also be caused due to severe conditions. Those results in physical damage to cells. Most common phenomena of cell death in a bioreactor is apoptosis, is also known as programmed cell death, which is regulated by a cellular cascade in response to nutrient depletion in cell culture medias, metabolite excretion, too much shear forces. During apoptosis, cellular DNA is fragmented and can be assayed to quantify apoptosis in a population of cells. It is important to avoid or hold back apoptosis in a cell culture to expand the period of cell viability and prolong protein production. Strategies that can work for it are manipulation of cellular environment by media, supplementation or the manipulation of intracellular environment using genetic engineering, supplementation with nutrients provide protection and used as a prevention in order to control the cellular environment, and setback apoptosis. Serum contains some factors that delays apoptosis. Therefore, the formulations that are serum-free make the cells more vulnerable to apoptosis. The next factor is glycosylation. Using animal cells is preferred for the production of biopharmaceuticals because they are capable of adding carbohydrate groups or glycans to the protein synthesized. Expression of appropriate genetic sequences control and direct the protein structures, whereas controlling the glycomics is still difficult. Glycosylation variations may be found in occupancy site or in attached glycan structures. The third system is the plant cell culture system. using plant cell cultures for producing human biopharmaceutical proteins has been under assessment for decades. Advantages of using plant cell cultures over mammalian cultures are cost effectiveness and absence of mammalian derived components in the production process. Growth process of plant cells can be controlled, which leads to high batch-to-batch reproducibility. Plant cell media lacks mammalian components which are vulnerable to the transmission of mammalian viruses. Plant cell systems don't bear the risk of infection by animal pathogens. Additionally, there is a natural barrier present in the plant cell cultures against mammalian pathogen contamination. Also, plant viruses cannot outspread in plant cell cultures because of the presence of plasmodesmata, which does not provide any route for virus movement. The factors laid down are important concerning safety and also reduces operational costs as compared to mammalian cell-based expression systems. Though proteins produced in plants and human proteins have some pattern of heterogeneity, still there are slight differences in glycosylation. Similar to mammals, plants also attach both nitrogen and oxygen-linked glycans, but the systems present in plants and mammals are not identical. Modifications in glycosylation pattern may not in particular affect the activity of protein, but is potential of generating immunogenicity in humans. Nitrogen glycosylation is the most important post-translational modification as 30% of all approved biopharmaceuticals contain nitrogen-linked glycans. Plant-derived nitrogen-linked glycans can be divided into three types: high mannose consisting of 5 to 9 mannose sugars in addition to the core, complex glycans and polymannose glycans derived from complex types but truncated in the plant vacuole. The complex nitrogen-linked glycans of plants are different from animals because they do not contain terminal sialic acids. As for an example, here in this diagram we can see the difference in the glycosylation patterns in plants and mammals. Now we will discuss the strategies for humanization of proteins. The strategies for humanization of protein N-GLYCOSYLATION in plants. One approach is to retrieve the expressed proteins in the ER by adding a carbon terminal tetrapeptide H or KDEL motif. The procedure involves formulating knockout of endogenous plant glycosyl transferases that transfer beta xylose and alpha fucose residues onto nascent proteins and engineering of the mammalian glycosyl transferase such as beta-galactosyl transferase or beta-N-acetyl glucosamine transferase 3 into host plants. Impact of the plant ooglycans in the stability, biological activity and efficacy of the therapeutic proteins has been less studied. Genetic engineering can be applied to avoid the plant-specific ooglycosylation. Here we can see with the help of this table the plant cell-based biopharmaceuticals developed till now. In another table, we can see the companies commercializing plant cell-based therapeutics with their products. Now, we will switch to the next system, that is the transgenic animals. Use of animals as bioreactors for the production of biopharmaceuticals adds significant value because of low cost implementation, production and scale-up, and it provides high output of synthesized proteins. Also, humans have existing phylogenetic relationship with other mammalian species, which allows animal bioreactors to synthesize recombinant proteins structurally similar to their original forms. It is difficult to separate human proteins from their animal equivalent, might be the drawback using animals as bioreactors. Particular attention is required for checking the animal pathogens that are active in humans should not be present in the protein preparations. Number one system is transgenic animal systems, and the possible systems for the production of recombinant proteins in transgenic animals include milk, blood, egg white, seminal plasma, urine and silk gland, insect and larva hemolymph. Now, we will study the different methods of generating transgenic animals, as also been illustrated in this diagram. Generating transgenic animals can be achieved by using microinjection and injecting DNA into embryo pronuclei, using lentiviral vectors or transposons, in vitro fertilization using sperm with a specific gene, transferring the foreign gene into pluripotent cells, afterwards generating chimeric animals harboring normal and transformed cells, and lastly, transferring the foreign gene into somatic cells used for the generation of cloned animals using nuclear transfer. As for an example, here we can observe the production of casein DNA in the milk obtained from a transgenic animal. And in the next table, we can see the companies and the recombinant products obtained from the transgenic animals. Last system to be discussed here is transgenic plants. Using plants for producing recombinant proteins is known as plant molecular farming or farming. Plants can easily be transformed and have significant potential to produce biopharmaceutical proteins and peptides. As discussed in the above sections, biopharmaceuticals or proteins produced in the cell culture systems are required to be purified from the culture supernatant. This purification step is an expensive process, while in case of plants, they can easily store proteins in the seed endosperm and can easily be extracted out. However, purification step is expensive, while many methods are developed to rise above this problem. One of it includes expressing proteins fused with oleosin. Also, in some cases, purification of protein is not necessary, like in the case of edible vaccines. Now, we will check the advantages of plants using as culture systems. Plants don't act as hosts for human infectious agents. Products derived from plants are less prone to be contaminated with human pathogens than the products derived from the animal cells. The source, that is, the plant leaves or seeds for making the recombinant protein, is potentially indefinite and as shown in this figure. Now, as a part of methodology for protein production in plants, the first step is selection of crop species. Plants that can easily be manipulated and also produces stable transgenics includes the tobacco, alfalfa, soya bean and other crop species like potato, tomato, banana, maize and lettuce. The next step is tissue selection. As we can see from the figure shown here on the screen, tissues for the production of proteins can be leaves, tubers, seeds, etc. Lastly, we can observe certain plant-derived pharmaceuticals that are listed here in the table. In the end, we can conclude the different platform technologies used for the development of biopharmaceuticals have different advantages and disadvantages. In the end, we can say the choice of using mammalian cell line systems is not restricted to their better production values and can be seen in recent trends. Around 2/3 of total revenue is produced by various marketed biopharmaceutical products. This shows a clear dominance of mammalian systems over their counterparts such as E. coli and yeast systems. In addition, the biopharmaceutical products manufactured using animal cell lines are more compatible and achieve higher purity, safety and potency levels according to regulatory standards. Biopharmaceuticals pose to be revolutionary in the pharmaceutical industry. According to global revenues, 10 biotechnological related products figured among the top 25 selling drugs in 2015, 4 of them produced by microorganisms. Biopharmaceuticals are marketed by leading pharmaceutical companies located in USA, Japan and Europe and comprise a narrow scope of treatment profile, with most drugs for the treatment and management of inflammatory diseases and cancer. With the continuous advancement in technology, innovative methods to improve the discovery, modification, production and purification of biomolecules are to be developed. Thank you.